So, before you know the difference between antibody and T-cell receptor, you must know the structure of antibody. An antibody is composed of mainly two chains called light chain and heavy chain. So antibody composed of two light chain and two heavy chain. This light chain can be of kappa or lambda type and the heavy chain can be of 5 type depending on the different constant region. And this antibody is also composed of two regions called variable region and constant region. So you can see a light chain contains one variable region and one constant region, and a heavy chain composed of one variable region and 3 constant region. So both the heavy and light chain actually composed of variable region and constant region, and this variable region is responsible for the binding of antigen, and rest of the constant region is responsible for the effector function of antibody. Then you have to know a term called hypervariable region in Ig molecules. Ig means immunoglobulin molecule. As I have already mentioned that the heavy chain composed of variable and constant region, and the light chain also composed of variable and constant region. This variable region is responsible for the antigen binding, but the whole region is not responsible for the antigen binding. Each variable region from heavy and light, they have 3 stretches of amino acid, approximately 10 amino acids, that form a region that is most variable. So it is called hypervariable. So this region is present on the variable region, so it is called hypervariable, because among the variable region, these 3 stretches of amino acids are most variable and they form a 3-dimensional structure that can bind with a antigen. So that region, this hypervariable region, is also called CDR, means complementarity determining region. Complement with the antigen, so it is called complementarity determining region. Now you can see there are 3 CDR called CDR1, CDR2 and CDR3. This plot is called Kabat-U plot because those 2 scientists discovered this fact. This is showing the variability of amino acid, and this is the number of or the position of amino acid. Now you can see that CDR1 and CDR2 and CDR3, among them 3, CDR3 is most variable among these CDR1 and CDR2. So in the variable region there are 3 CDRs like CDR1, CDR2 and CDR3. These CDRs are the hypervariable region of the variable region, and among this hypervariable region, CDR3 are the most variable region. You must remember this. BCR is nothing but membrane bound IgM and IgD antibody. This antibody also associated with some molecules Ig alpha and Ig beta. These 2 molecules are actually very important, and a B cell is also contain a molecule called CD19, and also a B cell receptor associated molecule can be CD21. It is a complement receptor that can bind with a complement protein called C3d that is attached with its antigen. Next, the structure of T cell receptor and associated signaling molecule. T cell receptor complex also composed of some molecules, associated molecules like CD3 and zeta, and you can see the structure of TCR. The T cell receptor is composed of alpha and beta chain. In this case also the alpha chain composed of variable and constant region, and the beta chain also composed of variable and constant region, as because the T cell receptor is also a immunoglobulin family. Structural difference between antibody and T cell receptor. Now the first is component. The immunoglobulin or antibody is composed of heavy and light chain, and the T cell receptor is composed of alpha and beta chain. The most common TCR. Why it is called most common TCR? Because T cell receptor is of 2 types called alpha beta, and there is another type of T cells that is very rare but also present called gamma delta. So most T cell receptor is composed of approximately 99.5% T cell receptor are composed of alpha beta chain. So the T cell called alpha beta T cell. But there are subtype of T cells that are called gamma delta T cells. They are very rare, 0.5%. The number of Ig domain, in this case heavy chain, 1 variable region and 3 or 4 domains of constant region. 3 for IgA, IgG, IgE, and 4 constant region for IgM antibody. The light chain always contain 1 variable region and one constant region. The T cell receptor is only composed of one variable domain and one constant domain. The number of CDRs in case of immunoglobulin is 6 because each variable region from heavy and light chain composed of 3 CDRs, so the total number is 6. And in case of T cell receptor, there is also 6 number of CDRs are present. The associated signaling molecules in case of antibody or B cell receptor is called Ig alpha and Ig beta, and in case of T cell receptor it is CD3 and zeta. So the next difference between the antigen receptors like Ig means immunoglobulin, antibody and T cell receptor. Now it is showing the difference or features of antigen binding. Antigen binding site made up of 3 CDRs in VH means variable heavy and 3 CDRs in variable light domains. So these 6 CDRs made a site where the antigen can bind. In case of T cell receptor, you can see it is also made up of 3 CDRs in V alpha and 3 CDRs in V beta domain. Here it is showing that macromolecules any nature like protein, lipid, polysaccharide. And I want to mention that immunoglobulin or antibody can bind with a soluble protein also or soluble antigen, but T cell receptor cannot bind with a soluble antigen. It can bind only peptide, means protein in nature antigen, but with MHC complex molecules. So T cell receptor cannot recognize other antigen like lipid, polysaccharide, and also cannot recognize soluble antigen or soluble peptide. It can only recognize peptide with MHC complex. Now another term is called antigenic determinant. It means the binding site of the antigen. In case of antibody, it can bind with linear and conformational determinant. Linear means denatured condition and conformational means alpha, beta sheet, any type of conformational determinants. T cell receptor can only bind with linear determinants of peptide. Now another important term called affinity of antigen binding, the strength of antigen binding. In this case, I also want to mention that this affinity is showing by or represented by Kd, mean dissociation constant, and this dissociation constant is inversely proportionate to the affinity. That means the lower the Kd, the higher the affinity. So you can see here the 10 to the power minus 7 to 10 to the power minus 11. So Kd in case of antibody is very low, so its affinity towards the antigen is very high. But in case of T cell receptor, the dissociation constant is 10 to the power minus 10 to the power minus 5 to 10 to the power minus 7. So it is low than the antibody. You must remember this. On rate and off rate. On rate means binding affinity or binding probability, and off rate means releasing probability. So here you can see that rapid on rate means the antibody can bind with a antigen with rapidly, and variable off rate, it can release the antigen variably. But T cell receptor bind with antigen slow rate, and it also release the